let's go ahead and get started. First of all, I intended to grade everything this weekend, and my weekend kind of got a little chaotic. So I did not get to everything. Most of your content checks are graded. I have maybe about 20 that were submitted on Google that I haven't gotten to yet. So if you don't have a grade, you will by the end of the day. I guarantee that. Test corrections, I have not done yet. I tried to prioritize the content checks, since that is information you will actually be tested on in the next exam, right? And then I will get you these content check 4s that are due today back to you before Wednesday, so you can use them for study. Another thing that was pointed out to me in my last class, that lecture recording for Friday, it is on YouTube, but after a certain point, the image stays the same even though the audio continues. I'm going to look at that. Great thing about teaching 2 lectures is I record 2 lectures, so I might just swap it out with one that's not corrupted, like this one seems to be. So hang tight on that video. I'll fix it today. Hopefully the other video is going to work better. All right. Any questions I can answer before we get started? All right, so on Friday, we went over the light reactions, right, and all the complexity there is to the light reactions. As we went through the light reactions, we filled out that graph, right, where we showed where the energy of the electrons moved, right? That graph is actually a really common graph to see in photosynthesis for the light reactions. It has a name even. It's called the Z-scheme, right? So I remember studying with my roommate back in my freshman year. We would study the Z-scheme, and we would always do like hand movements, and that helped us remember it. So if something like that helps you remember, great. It really deals with the movement of the electrons, and it's kind of in a zigzag pattern. Right? We have photosystem 2, which happens first, exciting our electron, right? So our electron has been excited. It's going to lose some of that excitement, lose some of that energy through the first electron transport chain, which means it has to be re-excited at photosystem 1, right? And once again, it's going to lose a little bit of energy through the second electron transport chain before it's finally accepted by NADP+ to create NADPH. All right, so light reactions produces NADPH, but we did not quite finish what happens here, right? There's still one more thing that happens, and that's the production of ATP. So we have this electron moving through these proteins in our thylakoid membrane. As it moves through, right, it's building up this gradient of hydrogen ions, and that gradient drives the production of ATP very much in the same way as it does in the electron transport chain in cellular respiration, right? Building up a gradient on one side, or specifically on the inside of our thylakoids, right? These hydrogen ions want to move with their gradient. The only way they can do this is by passing through ATP synthase. And as they pass through ATP synthase, right, ATP will be produced, right? We're going to phosphorylate ADP. So we're going to add a phosphate group to ADP to make ATP. All right, it's all about that gradient that has been formed. There's actually 3 different ways this gradient has been formed. The first one, I actually have listed as number 2 up here, but one of the ones that is the most obvious is exactly the way I just described it, right? As the electron moves through the electron transport chain, it pumps hydrogen ions across the membrane to get that buildup, right? Same thing that happens in cellular respiration. But there's also 2 other ways that are a little bit more subtle that adds to this gradient, right? The first is the splitting of water. So remember, this electron that moves through the light reactions has to be replaced, and it's replaced by an electron in water. So when water splits, right, we're getting oxygen, it's releasing an electron, but we're also getting some hydrogen ions, right? Hydrogen ions are being released on the inside of our thylakoid. thus adding to our gradient. The other way that we're adding to this gradient is by forming NADPH, right? So that final electron acceptor is NADP+, right? Turning into NADPH. This happens outside in the stroma, outside of our thylakoid. And we're reducing the number of hydrogen ions on the outside by incorporating it into NADP+, plus that hydrogen ion, to create NADPH, right? So we're using it up, making the concentration outside of the thylakoid even lower. All right, so 3 ways we can build up that gradient. Through the electron transport chain, through the splitting of water, and through the creation of NADPH, right? All leading up to that gradient so that we can produce a lot of ATP through our ATP synthase. I like this image. It's a very succinct image for the light reactions. It shows everything all in one area, what's going on. Might be a good image to help use in studying. So the third one, the production of NADPH. So we're using up hydrogen ions on a side that already has a low concentration, thus exacerbating the difference on either side of our membranes. All right, that is actually the last bit of the light reactions. All right, so 3 things have been made. We have produced oxygen through the splitting of water. And that water had to be split to replace that lost electron. The final electron acceptor is NADP+, which combined with an electron and a hydrogen ion, we get the production of NADPH. Right? And this is a electron carrier molecule, just like NADH is. And then we're producing a lot of ATP. Right? ATP is being produced via ATP synthase. So we're producing oxygen, which is kind of a byproduct. It's actually like a waste product for the plants. They're going to get rid of that oxygen. But then we're producing these 2 other molecules that are energy-containing molecules. And they're going to be important for the second part of photosynthesis. Right, before we move into that second part of photosynthesis, though, I thought it's a Monday. It's been a weekend since we've learned about the light reactions. Let's watch a review video. I've linked it on the slide because I couldn't physically embed it for whatever reason. So I'm going to move over to that YouTube video now.
The thylakoid membranes contain specialized molecules that work together to perform the light reactions. Light is absorbed by protein pigment complexes called photosystems. There are 2 photosystems, photosystem 1 and photosystem 2. The photosystems transform light energy to chemical energy by exciting and then shuttling electrons from molecule to molecule in a chain-like fashion on the thylakoid membrane. This process is called an electron transport chain. Let's take a closer look to see how this process works. First, photons of light hit chlorophyll, a light-absorbing pigment in photosystem 2. Electrons in the chlorophyll are excited to a higher energy level. The excited electrons are passed to an electron carrier. Meanwhile, water splits and releases electrons. These electrons replace those lost at photosystem 2. The byproduct of this reaction is oxygen, which is eventually released into the air. The other products are protons, or hydrogen ions, which are released into the inside of the thylakoid, or lumen. The excited electrons move to the cytochrome complex. Some of the energy from the electrons is used by the cytochrome complex to transport additional protons into the lumen. The second electron carrier, a protein inside the lumen, receives the electrons and passes them to photosystem 1. These electrons have now lost most of the energy they gained from light in photosystem 2. Photons of light hit chlorophyll in photosystem 1 and excite the electrons again. The electrons are then passed to the third electron carrier. Finally, these electrons are either recycled or they interact with an enzyme and NADP+, the final electron acceptor of the light reactions, to form NADPH. Some of the energy from light is now stored in the reduced molecule NADPH. Some of the energy released from the transfer of electrons established a proton gradient across the thylakoid membrane. Protons that accumulated in the lumen diffuse into the stroma through an enzyme called ATP synthase. ATP synthase uses the potential energy of the proton gradient to combine ADP with inorganic phosphate to form ATP. In this way, the potential energy is transformed into chemical energy stored as ATP. ATP and NADPH now have stored energy from the light reactions. This energy can be used in the Calvin cycle. This light-driven electron transport chain is usually continuous in the presence of sunlight. It encompasses a series of chemical reactions that involve light absorption, energy conversion, and electron transfer carried out by the photosystems and other enzymes on the membrane of the thylakoids.
All right, so this video goes in just a little bit more detail than what we talked about in terms of like the number of electron carriers. But overall, does a really good job of explaining it and then physically showing how this process all happens at once, right? So it's not one electron at a time, it's actually an electron moving with another one coming in right after it, another one coming in after that one, right? It's a constant thing. All right, before we leave the light reactions, I did have a question posed to me about where is the antenna complex in the light reactions, right? Well, the photosystems, right? The photosystems are actually made up of an antenna complex and a reaction center. So if I were to ask you this question, where is the antenna complex in the light reactions, you would tell me the photosystems, right? Because photosystems are made up of antenna complexes and reaction centers. All right, any questions about the light reactions? All right, we have produced oxygen, it's kind of a byproduct, a waste product. It's been released into the air. But we've also produced NADPH and ATP. These two molecules are going to move into the next part of photosynthesis that we call the Kelvin cycle, also called the dark reactions, because it doesn't directly need light. Kelvin cycle, I think, is a little bit easier than the light reactions. There's fewer steps, all right? So we got the hard part done and over with. We're going to go on to the easy part of photosynthesis now. The Kelvin cycle is where we actually take CO2, right? So CO2 is entered into the leaves via the stomata, right? It's now entered into a mesophyll cell and specifically into the chloroplast, where it's going to be utilized in this cycle. The CO2 is going to be incorporated into other molecules to eventually make glucose, to make sugars, right? This is actually a process that requires a lot of energy, right? Which is why the light reactions are so crucial. The light reactions provided all those energy molecules, ATP, NADPH. If we look at it in terms of numbers here, for every 6 molecules of carbon dioxide, we need 18 ATP and 12 NADPHs. Right? That's a massive amount of energy needed for only 6 molecules of CO2. All right, so this Kelvin cycle, coming in, oxygen is, or CO2 is coming in, combining with a molecule, eventually making sugars. Right? The direct product of this cycle isn't actually a sugar. It's actually glyceraldehyde 3-phosphate, which should sound familiar, right? The same molecule made an appearance in glycolysis. Right? That is the direct product, And then once this product is made, there's an extra step to turn it into glucose. All right, so big picture, CO2 is coming in, we're making sugars, right? This requires a lot of energy, and we are getting this energy from the light reactions. All right, the Kelvin cycle, it has a lot of steps, but again, we're going to focus it down to just the main portions. I'm not going to have you memorize every single molecule in this entire cycle, right? So if we look at it from a broad perspective here, right, this image does a good job of breaking it down into its three phases. We can see on this image there's three colors kind of in these triangle patterns here, right, that all each indicate a phase. Phase 1 is carbon fixation. Phase 2 is reduction and carbohydrate production. Kind of rhymes a little bit, might help you memorize that. And then the last phase, right, the third phase, right, is regeneration of RUBP, which is a molecule that we'll see when we break this down. So we're fixing carbon, we're making sugars, we're regenerating a molecule so that the cycle can continue. All right, we will circle back to this image, but let's break it down into what specifically I would expect you to know about the Kelvin cycle. So, phase 1, carbon fixation. So this is the part of the cycle that incorporates CO2, right? We have CO2 coming into the cycle and combining with this molecule called RUBP. Now, an enzyme facilitates this combining, right, the combining of CO2 with RUBP, and that enzyme is called Rubisco. And we will come back to Rubisco at the end of this lecture to see why it's actually a really important enzyme. Right? So CO2 combines with RUBP to make 3PG, or 3-phosphoglycerate. I know everybody likes to memorize these shortened versions of molecules, but I would say for this one, 3GP, it might almost be easier to memorize the full thing because it sounds very similar to G3P, right? G3P, glyceraldehyde 3-phosphate, versus 3PG, which is 3-phosphoglycerate, right? I think these shortened versions can be more easily mixed up than the long versions, so keep that in mind. Right? But 3-phosphoglycerate molecules, right, actually 12 3-phosphoglycerate molecules is made for every 6 CO2 that enters into this system. So once CO2 combines with RUBP, we make 3-phosphoglycerate molecules. And that is the first step, right? That is all that I would expect you to know for the first step. So we've incorporated CO2. We've used that CO2 up. We've produced molecules of 3-phosphoglycerate. That phosphoglycerate will enter into the next phase here. Now before I move on, I'm just going to give you some extra time to write, so I'll give you a little foreshadowing of what we're going to talk about in the next couple of slides. But this 3PG, it has that 3 in there because it contains 3 carbon atoms in that molecule. Plants that utilize this are called C3 plants because of it. All right, 3-phosphoglycerate molecules are going to enter into stage 2, reduction and carbohydrate production. This is where we are actually going to see glyceraldehyde 3-phosphate made, which will eventually make sugars. So we have our 3-phosphoglycerates, right? We're going to go through a series of steps, which I'm not going to have you learn each individual step, right? But in the process of turning 3-phosphoglycerate into glyceraldehyde 3-phosphate, right, we are using a ton of energy. We're using up a lot of ATP and a lot of NADPH. Right? We're using up those molecules that we produced in those light reactions to turn phosphoglycerate, 3-phosphoglycerate, into glyceraldehyde 3-phosphate. Now again, if we look at the numbers here, right, we're talking about 6 CO2 combining with 6 RUBP molecules to make 12 3PG, right, 3-phosphoglycerate. 12 of these 3PG will turn into 12 glyceraldehyde 3-phosphate. Now, only 2 of these are actually going to leave this cycle to create sugars, to create glucose. The other 10 are going to continue on to this next cycle. Right? So even though we produce 12 glyceraldehyde 3-phosphates, only 2 of them are going to be used to make glucose. So this is a very energy-intensive step here. All right, so we have 10 of these molecules staying in our cycle, moving on to our next step, which is the regeneration of RUBP. All right, phase 3, regeneration of RUBP. Just like we saw in the Krebs cycle in cellular respiration, where we had to regenerate a molecule, right? We had to regenerate oxaloacetate. Now we have to regenerate RUBP so we can continue the Kelvin cycle, right? So 10 of these glyceraldehyde 3-phosphates are staying within the cycle. They're going through a series of reactions that use up even more ATP, right? And as a result, we're creating more RUBP that can then enter back into the carbon fixation cycle and start it all over again. This is a nice step. You don't have to memorize any new molecules here. All right, that is the Kelvin cycle, right? So it's much less intensive than the light reactions. Great question. This actually occurs in the stroma, right? So the stroma is that area between the thylakoids and the membranes of our chloroplasts. So we're within the chloroplasts, but we're outside of the thylakoid, all 3 phases. So light reactions happen across the thylakoid membranes. The Kelvin cycle happens in the stroma inside of our chloroplasts. All right, so if we were to recap here, we have CO2 combining with RUBP using this enzyme called rubisco and producing 3-phosphoglycerate. 3-phosphoglycerate, through a series of steps that are very energy intensive, produce glyceraldehyde 3-phosphate. Of the 12 glyceraldehyde 3-phosphates produced, 2 of them leave to make glucose. 10 of them stay on for the 3rd phase, again energy intensive here, to regenerate RUBP. All right, let's put this into practice. No, I thought I had a practice slide next. I don't. My last slide here is just, it's to reiterate rubisco. Rubisco is that enzyme that helps CO2 be incorporated into RUBP. And it's important. It's very important. We're going to see that after we do a group review here. It's found in every single photosynthetic organism that we have studied. And it's thought to be the most abundant enzyme on Earth. Right? That's how important this enzyme actually is. Because it's such an important enzyme, it would probably be good to know the name of this enzyme going into the exam on Friday. All right, now let's do a review. I have been known to put this exact image on exams, so this is a great slide to study off of. I want you to fill in the blanks of the Kelvin cycle.
It's the by beyond us.
So the second phase, keep on.
It's 1 3 BPG. Yeah.
I counted 6 bonds.
What number? The bottom Oh, it's the R.
What is like the end I actually put it in.
Who?
That's what's created here before it's further reduced to create heat. Yes. So don't focus on that one. Focus on it's an intermediate. It would be if I were to put it in the cycle, it would happen right here.
That's
Once
6
an
bonds.
aggregate enters into together, it will convert it into that was an end. That's something where if I were to go back and edit the slide.
You got the 6 RUP.
I already had this.
So it makes, this makes full. So, really the product. This is going on.
Yeah, it's depending on, I didn't talk about it. Well, one new partner says that we have all these,
All we're doing is. So the left side is.
Okay, I already.
Okay, stage number one, let's go through a review and then I'll take any questions we might have. What is that first phase called? Carbon fixation. What happens in carbon fixation? What molecule enters? CO2. What molecule does it combine with? RUBP. And what does it create? 3-phosphoglycerate. 3PG. Yes. What about that second phase? What is it called? That's reduction in carbohydrate production. And what is made? It already tells you on here that G3P is made, glyceraldehyde 3-phosphate is made, but what does it go on to make? Glucose. Right, and then we enter into that last phase, right? We have to regenerate our molecule of RUBP. All right, are there any questions on this cycle? One of the slides mentioned biphosphoglycerate, right? That was an intermediate that we would find in the second phase. I don't expect you to know that one, right? Everything on this slide is essentially what I would expect you to know. And remember, we also have our enzyme Rubisco. I would expect you to know that as well. All right. Any other questions before we move on to our next topic? I have shown this exact image on slides. I have shown modified versions of this image, or excuse me, on exams. So this is a great one to know. All right, so far we have learned the normal way that photosynthesis will occur, right? The way we have just learned is like 90% of the plants on planet Earth will go through photosynthesis in this way. However, Earth varies, right? What's happening in Minnesota right now? I mean, it's really, really cold this morning versus what's happening in other parts of the U.S. or the world, right? I think a student in my other class told me it was 94 degrees in his hometown in Texas, right? There's a lot of variability in the environment, and the plants have to compensate for that variability, right? There might be different light intensities. Think desert sun versus like what noon is in Minnesota or closer to the Arctic Circle. Temperature, which I mentioned, but also water availability, right? We tend to have plenty amounts of water where we live, right? But again, think like the desert Southwest, right? There's not a lot of water in like the Mojave Desert, right? Plants have to contend with these variable conditions, right? And they do that by kind of balancing how much water they're going to lose versus how much CO2 they're going to take in. So if we think about their stomata, right, the pores, the opening on the leaves, this is where the CO2 comes in, right? Stomata, they're normally open during the day when photosynthesis is happening and closed at night, right? Again, this is like 90% of the plants, this would be normal. However, when the stomata are open, plants are actively losing water, right? So think about plants that live in the desert. They don't have access to a lot of water. They don't want to lose it all when they open their stomata. So they have to balance water being lost when the stomata is open versus CO2 coming in, right? Now they could just close their stomata, right, and not photosynthesize. Well, that kind of works, but not for long periods, right? So if the stomata are closed, they're not losing water, but that also means water's not flowing through the plant itself, right? It's a whole other process, transpiration, that we're not going to get into on this class. If you take organismal biology, you'll talk about this process, right? So if they totally close it off for long periods of time, they could risk dehydration. So it's all about this balance between water loss and gas exchange, bringing in carbon dioxide and even getting rid of the oxygen that's produced with photosynthesis. All right, so we close the stomata, we're stopping the delivery of CO2, right? Thus, we're stopping photosynthesis, specifically the Kelvin cycle, which uses that CO2. Now, the light reactions can still continue, right? They're not directly using CO2. They can still continue on, right? But what's going to happen is that, during the light reactions, water splits to replace a lost electron, and that creates oxygen. Oxygen is then going to build up. And this is a problem, because this can lead to something called photorespiration, where instead of using CO2, right, the enzyme Rubisco is going to start using oxygen instead. Right? So it's this reverting of Rubisco to using oxygen instead of CO2. That is photorespiration. This is not efficient. Plants do not want this to happen. All right, let's look at why this is not efficient. All right, so if we look at the role of Rubisco normally, right, what 90% of plants on Earth, they tend to do photosynthesis in this way. RUBP combines with CO2 via the enzyme Rubisco to create 3PG, 3-phosphoglycerate. That's what normally happens, right? We are making this molecule 3PG, 3-phosphoglycerate. It has 3 carbons in it. Thus, plants that go through this process, we call C3 plants. 90% of plants on Earth are C3 plants. Right? I put an example of an oak tree out here, but I could really point outside the window, and most of the time I would be pointing at a C3 plant. Right? So this is what occurs normally, using CO2. Now, the plants that have to live in these conditions that are harsher, they don't have as much water, they have to balance when their stomata are open, they have to find ways to minimize photorespiration, right? Which is what will happen in C3 plants if they don't have an alternative strategy. So when oxygen builds up and photorespiration occurs, what we see happening is that RUBP is now going to combine with oxygen instead of CO2, right? So Rubisco can also combine oxygen with RUBP. This only yields 1 3-phosphoglycerate, right? Whereas the normal reaction yields 2, right? So we're yielding less. Plus, we're also producing this other molecule called 2-phosphoglycolate. this other molecule that's produced has to be further processed, right, to make it into a 3-phosphoglycerate. This takes yet another CO, or this releases yet more CO2 and uses more energy. It's not efficient, right? So we're using more energy to basically get to the same end result, right? If we look at Rubisco, that enzyme, it prefers to use CO2, right? We would say that the affinity for CO2 is 10 times better. It much prefers to use CO2. But if there's not a lot of CO2 and the oxygen concentrations are greater than the CO2, they can use it, right? So basically, photorespiration is occurring when we don't have enough CO2 coming into the plant. And photorespiration is bad because it's not efficient. It uses up more energy. So if I were a C3 plant and I had to close my stomata or risk dying, right, this is what would happen inside of my cells. I'd have to use up more energy to get the same result. All right, well, let's do a review before we move on here. And then we'll briefly touch on some variations before we get to the content check, which is a little easier this week.
No, yeah, because when the temperatures are high, water then it evaporates faster. It's called
How's it going over here? Yes. Yes.
Oh, it tastes awesome.
The second one?
Instead of CO2.
Eating the CO2,
I
which
like bioactive.
So it's creating water. Okay. Okay. We have another question? Yeah. Yes. Oh, yes. Yes, you'll put it that way. Yep. It's okay. Oxygen is what breaks down to hormones. Water, yes. Water gives away the electrons. Just like
And then we'll have 2 pipes. So instead of having
all right, so what is the problem with Rubisco when temperatures get really high? It gets really hot outside.
Kelvin cycle stop.
Why does the Kelvin cycle stop?
Because there's no CO2 used to make Rubisco.
Yeah, that because the stomata have closed, right? Stomata closes, which means there's no more CO2 coming in. The Kelvin cycle can stop, right? But what if, right, Rubisco, instead of using CO2, starts to use oxygen? What is this called? Photorespiration. So unless plants have an adaptation to overcome, right, photorespiration is going to occur, right? So what is the trade-off between, as it tries to gain CO2 to synthesize sugar? What is the trade-off for the plant? It'll lose water. Yeah. Stomata are open, they're going to lose water, but they're gaining the needed CO2 needed for the Kelvin cycle. All right. So in these hot and dry climates, plants need a way to minimize photorespiration, and there's two slight variations on photosynthesis that plants can undergo, right? They can be a C4 photosynthesizer, or they can be a CAM photosynthesizer, right? So in C4, they're going to separate some processes in space, right, or in a spatial separation, so between different locations. Versus CAM, we're going to separate processes in time, right? And the whole reason these separations occur is because they need a way to concentrate that CO2 so that Rubisco will not use any oxygen that is produced. All right, we're going to break down these just a little bit further. Right? So C4 photosynthesis is what we're going to look at first. This is the spatial separation of processes. So if we go back to our leaf structure, we know photosynthesis happens in mesophyll cells. But in C4 plants, they have the addition of another type of cell we call a bundle sheath cell. Right? If we look at our cross-section of a leaf here, again, most of these on the inside, all of these green cells are mesophyll cells. But there's going to be a layer surrounding any veins inside of the leaves, a layer of cells specifically called bundle sheath cells. So part of the reaction of photosynthesis will happen in the mesophyll cells, and then another part is going to happen in the bundle sheath cell. So CO2 is entering into the plant. It comes into the stomata. It enters into the mesophyll cells. It enters into the chloroplasts, right? This is all happening in the mesophyll cells. But now we have an extra step where we have to basically move CO2 into another type of cell. Now, in order to understand this, I am going to go through the process. Highly unlikely that I will ask you the steps of this process because I want you to think big picture when we're thinking about variations of photosynthesis. But the only way to understand this is to literally look at that process. So, in C4 plants, when CO2 comes into the mesophyll cell, instead of combining with RUBP, it combines with this molecule that we abbreviate as PEP and uses the enzyme PEP carboxylase to do this, right? So it's combining with a different molecule at first. It's creating oxaloacetate, right? Again, that should sound familiar. We've heard about this when we talked about the Krebs cycle. Oxaloacetate will convert to malate, and malate is going to move into the bundle sheath cell. So you can think of oxaloacetate and malate as kind of like a shuttle bus for carbon dioxide. Because once it gets into that bundle sheath cell, it's going to release its carbon dioxide, right? And that carbon dioxide will go through the Kelvin cycle just as we have learned, right? Now it's going to combine with RUBP using Rubisco. Malate, once it gives away that CO2, turns into pyruvate and gets regenerated into PEP, and this process can happen over, right? So basically, we are spatially separating where things occur. By having the Kelvin cycle happen in the bundle sheath cells, and by having malate shuttle in carbon dioxide, it ensures that in the cells where the Kelvin cycle happens, we're always going to have plenty of CO2. All right, so spatial separation again here is key. So again, if we're thinking big picture, I'm not so worried about the little molecules, right? But rather worried about that you know that it happens in a mesophyll cell and a bundle sheath cell. CAM plants, on the other hand, have a separation in time, right? So CAM stands for crassulacean acid metabolism. It was named after the family of plants that this was first discovered in. And what CAM plants do is they actually open their stomata at night and close them during the day. Yeah, they're kind of like nocturnal plants, right? During the night, when the stomata are open, they're going to take in as much CO2 as they possibly can. That CO2 is going to be converted into malate, right? Malate converts into malic acid, and it's stored in the vacuole, right? So they're trying to store as much CO2 as they can at night. Now, during the daytime, right, the stomata will close. But that's not a problem because they've stored up that carbon dioxide, right? They're now going to use that stored up carbon dioxide for photosynthesis, right? It's going to leave the vacuole, it's going to enter into the chloroplast, and photosynthesis will occur as normal, right? So by closing their stomata at night, or by closing their stomata during the day, right? Now they're not losing water due to hot temperatures, right? But because their adaptation to store CO2, they're also not photorespirating, right? So it's a clever way to kind of get around photorespiration. All right, but again, in big picture, C4 versus CAM plants. All right, well, first of all, C3 plants, those are plants that go through normal photosynthesis, most of the plants on Earth. C4 plants separate processes in space, right? So mesophyll cell versus bundle sheath cell. CAM plants separate processes in time, opening stomata at night instead of during the day. All right, big picture again, separation in space versus separation in time. I kind of reiterated this already, and this slide you've seen before. I'm going to keep going so we have enough time for the content check. I was going to have us do a review, but again, to get to the content check, I made it on your own. These are good questions to ask as you're studying. Right, scientific literacy assignment, remember that's due on Wednesday, and we'll have an exam review, exam on Friday. And as you're studying, my biggest tip here is just write things out, all right? Draw it out. Put it into your own words, right? Study out loud with a partner. There's a lot of processes to know here, lots of molecules to know. Not nearly as many molecules as if we had looked at things in more detail, right? It could be worse. I have simplified it, right? Let's get on to the content check, though. Content check, I am asking you, where in the mesophyll cell in a C3 plant, so again, normal photosynthesis, are each of these processes happening? And I want you to be as specific as you possibly can to the location. Now, I told you this, but I forgot to tell my other class where the Kelvin cycle happens, so I'm going to give you this answer. Kelvin cycle, like I said, happens in the stroma. Right? Stroma of the chloroplast. That's as specific as you can go for the Kelvin cycle. The other three, you should be able to come up with an answer. Again, be as specific as you can. Once you're done, if you have a paper copy, I actually remember to stapler today. I'll make a pile up front. Otherwise, submit it online.